Hello students. Uh, today today I am going to deal with the student of MSc2 with the topic of cell growth and its bioproduct formation and its kinetics. Today I will deal with the growth pattern and kinetics in batch culture, fed batch culture, continuous culture. So we all have to know about what is culture, cell culture. In industrial biotechnology, we use bioreactor for a growth of cells and many pharmaceuticals compounds, hormones, enzymes by in vitro condition by the use of bioreactor. By giving some inoculation, bacterial inoculation in proper media. First of all, we all have to know about which types of which type of cultures are there in bioreactor. Mainly two types of cultures are formed in bioreactor. Number one is batch culture, in which cells are nurtured in a fixed volume of the medium until growth ceases. Means in the batch culture, the quantity of medium is fixed. Means a quantity of medium and inoculum is fixed. And at the particular time, the growth is at the maximum point. There it will cease. Second is continuous culture, in which cell growth is maintained by continuous replacement of sterile nutrient. In continuous culture, the medium is renewed in regular time interval, and the process is continuous. By this process, we can distinguish the growth cycle of the bacteria, as all we know that the bacteria and microorganism have particular pattern of growth cycle. So we are used here inoculum of bacteria, which is pharmaceutical capable of express some enzyme and antibiotics. So the pattern of growth in batch culture and continuous culture is step by step in lag phase, log phase, and stationary phase. Here is a diagram where you can see the sinusoidal graph where we can see the log phase. First one is lag phase, log phase, and stationary phase. First, we have to know about what is lag phase. The lag phase is a period of zero growth when cells are first inoculated into the growth medium. The length of the phase depends on the type of cell and their metabolic state in the lag phase. During inoculation, it is a period of adaptation which the cells recover from trypsinization, reconstruct their cytoskeleton, secrete matrix to aid attachment, and spread out on the substrate. Second phase is log phase. In log phase, the exponential growth phase is a period of exponential growth phase is a period of continuous cell doubling. Animal cells normally exhibit a doubling time of between 15 to 25 hours. The length of the log phase depends on the seeding density, the growth rate of the cell, and the density at which cell proliferation is inhibited by density. Third is stationary phase. In stationary phase, is a period after growth when there is no change in the culture cell density. The phase occur when the nutrients have been depleted or inhibitory metabolites have accumulated in the culture. Here we can see the growth curve of the growth curve in the batch culture. First phase is lag phase, second phase is log phase, and third phase is stationary phase. In the first phase, that is lag, here is the inoculum is given to the medium, and here the adaptation of microorganism occur. Here no replication or multiplication of bacteria microbes occur, only adaptation is occur in this phase. After that, the log phase. In the log phase, the doubling of the microorganism or animal cells are occurred. In this phase, the maximum growth of microorganism occur in the bioreactor, and the doubling time is exponentially high. After this the stationary phases occur. In stationary phase, the confluency in the exponential last phase, confluency occur in the medium. That's why no medium is left, because it is a batch culture. Particular amount of medium is given. No replacement of medium is occur. That's why here in the last exponential phase, the confluency is occur. The medium is utilized by microorganism. After that the stationary phase occur, where the confluency is maximum, and cell microorganism going to death because of the lack of nutrient and toxic presentation in the present in the medium. Here in the exponential phase, in the stationary phase, here subculture is occurred in the continuous culture and batch culture. This is the growth curve of the microorganism in the batch culture, or the kinetics of growth curve in the batch culture. Thank you.